Bioinformatics is an interdisciplinary field that develops and uses methods and software for analyzing and understanding biological data. Bioinformatics can be applied to understanding molecular data in a wide variety of ways. This includes DNA sequencing, which allows scientists to see the actual nucleotides that make up a DNA molecule, assembling entire genomes, and comparing DNA sequences to determine evolutionary relationships among organisms. DNA Subway is a simple and user-friendly website that ties together key bioinformatics tools and databases to assemble gene models, investigate genomes, work with phylogenetic trees, and, most importantly for this video, analyze DNA barcodes. Each subway line in the map contains a series of stations or analysis steps. DNA Subway can be used to analyze your own data, or you can practice using the vast library of sample data. In this video, we're going to talk about the blue line, determine sequence relationships. Once you've made your DNA Subway account and logged in, click the blue square to create a new project. Select CO1 under barcoding. The CO1, or cytochrome oxidase subunit 1 gene, is a mitochondrial gene that's commonly used as a DNA barcode among animals. CO1 mutates fast enough that sequences can be used to distinguish closely related species, since it mutates at roughly the same rate that species evolve. You're going to be analyzing and publishing DNA barcodes, or CO1 sequences, for the Tucson Bee Collaborative to document the incredible genetic diversity of our native bees. However, in this demonstration, I'm going to be working with sample DNA from DNA Subway of 5 fly specimens, 1 horsefly, and 4 mosquitoes. The sequence viewer allows you to see the nucleotides that make up your DNA sequence. If you click this button, you can see the DNA sequence chromatogram, or the visual representation of a DNA sample from a sequencing machine, and the computer's interpretation of the sequence. The 4 nucleotides that make up DNA, adenine, thymine, guanine, and cytosine, correspond to the A, T, G, and C in the sequence, each of which has a different color in the chromatogram. You'll also see ends in your sequence, especially at the beginning and at the end. Ends represent ambiguous nucleotides and are used when the true nucleotide at that base position is unknown. The height of each peak corresponds to the concentration of a single nucleotide at that position. Thinking back to our PCR reaction, remember that we amplified millions of copies of our target gene. When the sequencing machine reads these copies, the more agreement they have that there's a specific nucleotide at that position, the higher the peak. And above each peak is a confidence bar that shows the quality of your peak. Clicking the sequence trimmer will automatically trim the low-quality sections at the beginning and end of our sequence. In the pair builder, we're going to designate the forward and reverse reads of our samples. But what exactly does that mean? For each specimen, you have 2 reads, forward and reverse, each of which will have a unique file name. In the process of DNA sequencing, both of the complementary strands of DNA get sequenced, and each has their own sequence read, one of which is our forward read, and the other is the reverse read. Since DNA sequencing and DNA replication move in opposite directions, the reverse read is simply a reverse complement of our forward read. Once they're in the same orientation, the bases are put in reverse order and switched to their complementary nucleotides. To designate a forward and reverse read, simply click where it says F or R to switch it. After this, we're going to pair our forward and reverse reads for each specimen. To do this, click the check boxes next to the forward and reverse reads that you'd like to pair. Make sure to save your pairs before closing the Pair Builder window. Once you have your paired reads, click Consensus Builder. We have our forward read up here, and below it is the reverse complement of our reverse read. The consensus sequence shows the most common nucleotides at each position in both the forward and reverse reads. In the perfect scenario, the forward and reverse reads would be identical. However, due to computer or sequencing errors, sometimes it's just not the case. In our sequences, mismatched base pairs will be highlighted in yellow. The computer selects the nucleotide with the higher quality score to appear in our consensus sequence. Using the sequence trimmer, DNA Subway automatically cut off the low-quality parts of our sequence at the beginning and end. Now we have to do it by ourselves. To do this, click the button that says Trim Consensus and click the nucleotide up to which you would like to trim the consensus sequence at the beginning and at the end. After trimming our consensus sequences, we will compare these to published sequences on GenBank using a feature called BLAST to see if we can identify the species that our DNA sample came from. Based on how similar our CO1 sequence is to sequences in the database, we can effectively identify our specimens based on DNA. The results of our BLAST search show that our horsefly is most likely Calliphora vicina, and our mosquito specimens are most likely Aedes vexans, Aedes albopictus, Culex erraticus, and Culex territans. This brings us to the end of the video talking about the DNA subway blue line. Thank you so much for watching.